This is part 2 of the lectures on prokaryotes. We left off discussing how prokaryotes reproduce, and their prokaryotes, which remember include bacteria and archaea. They are organisms that are unicellular and they do not have a nucleus. They reproduce asexually by cloning themselves in a process called binary fission. However, there are 3 ways that there can be some genetic variation introduced into new bacteria, and the 3 ways are transformation, transduction, and conjugation. So first we'll look at the process of transformation. In transformation, the cell takes up prokaryotic DNA directly from the environment. So let's say, for example, that this green circular DNA is a plasmid, because that's the definition for a small circular piece of DNA is a plasmid. And that little small piece of DNA came from a bacterium that died, so a dead bacterium that is in the same environment as this bacterium with the purple genome. This is the one single circular chromosome for the bacterium here on the top of the picture. So the plasmid can actually enter this bacterium, and then it can incorporate itself into the genome of this bacterium. So in the future, every time this bacterium divides, it will contain those additional genes that the plasmid gave it, okay? And then for transduction, that happens through a virus. There we go. All right, so say this bacteriophage virus, notice the shape. It anchors itself and attaches itself to the bacterial cell wall and plasma membrane, and then penetration, it injects its DNA. So anytime the DNA comes from a virus, those genes can be viral genes, but they can also be genes from another source. The virus can be used as a vector to transmit genes into the bacterium that are actually bacterial genes. But the process of transduction always involves a virus, a bacteriophage. And so it does the same thing as transformation, except the DNA comes from the bacteriophage rather than a plasmid DNA, okay? And in conjugation, what happens is you have a bacterium on the left, which would contain a plasmid, and it can line itself up with another bacterium that doesn't contain a plasmid, and they can line themselves up and connect to each other through the sex pili. The sex pilus will connect these individual bacteria and allow that plasmid to enter the second bacterium. And now every time it divides, it will have those additional genes. So those are 3 ways that new genes can be introduced into bacteria. And then variations also occur in bacteria from just simply every time the bacterium undergoes binary fission, there can be mutations in the DNA code, just regular mutations during DNA replication. Okay, so now here we have an image of the relationship between the 3 domains of living things. There, from all of the evidence we have, there was a universal ancestor, which was a single-celled organism that gave rise to an organism that gave rise to all the bacteria, which are prokaryotes, and then an organism which gave rise to the eukarya and also the archaea. So the only 2 domains that are prokaryotes are actually archaea and bacteria. And they differ in their cell membranes and some characteristics of their cell wall, as well as some other characteristics like their ribosomal RNA But anyway, if you look at the bacteria, we won't talk that much about the different groups of Archaea, but we will look at the different groups of bacteria. We'll learn the proteobacteria, the chlamydias, the spirochetes, cyanobacteria, and gram-positive bacteria. These are the same groups that are in your lab manuals for this week. Okay, differences in the phospholipids between bacteria, eukarya, and archaea. So we know that eukarya, our cells, in other words, human cells, have a phospholipid bilayer. Okay, so this is showing that bilayer. This is showing phospholipids that are lined up with the tails facing. But in archaea, it's a single layer where the two phospholipid bilayers have fused. So there's linkage to where it makes it, there's a linkage between the two bilayers, making them a monolayer, okay? Differences in the cell walls, you need to know this. Bacterial cells, let's make sure I can write. Bacterial cells cell walls contain peptidoglycan. Archaean cell walls do not have peptidoglycan, but they may have pseudopeptidoglycan, which is similar chemically to the peptidoglycan layer in bacteria. They may have polysaccharides, glycoproteins, or even protein-based cell walls. So for bacteria, the cell walls always contain peptidoglycan. And we look at a lot of bacteria, but not all, can be divided into groups based on their gram stain. And this is something that we do and we have done in lab this week, or are doing in lab this week. If you look on the left, gram-positive bacteria has a very thick peptidoglycan layer in its cell wall, and gram-negative bacteria has a very thin peptidoglycan layer in its cell wall. So the thick peptidoglycan layer of the gram-positive bacteria allows it to absorb a purple stain called crystal violet and retain that stain. Oh, this is going to be ugly. Sorry. This word is purple. So the gram-positive, sorry, I really apologize. But the gram-positive bacteria will stain purple under the microscope. The gram-negative bacteria, when you do this step called the decolorizing step, where you add ethyl alcohol, that ethyl alcohol is going to wash off the purple dye from the gram-negative bacteria. Their little skinny, thin layer of peptidoglycan will not hold in the purple dye. So you have to use a different dye, which is called safranin, and it will give the gram-negative bacteria a pink color under the microscope. So gram-positive will look purple and gram-negative will look pink. And this is one of the ways that we can help to learn or discover the identity of our bacteria. We can identify them based on, whether they have a positive or negative gram stain. Structural differences and similarities between bacteria and archaea. They both have prokaryotic cells. So this is the same. Okay, in bacteria and archaea, the cells are prokaryotic, meaning they do not have a nucleus. They have DNA that's located in a nucleoid region. Cell morphology, which just means the structure, is variable in both. The cell wall contains peptidoglycan in bacteria and does not contain peptidoglycan in archaea. So that's an important difference. It was supposed to be an asterisk or a little snowflake, but it's ugly. But anyway, the bacterial cell membrane, the plasma membrane, which is actually inside the, it's underneath the cell wall. So if you look at bacteria at a bacterium, I thought maybe, sorry, I'll just draw it. If you look at a bacterium it's going to have a plasma membrane, and then after that it's going to have a cell wall outside of the plasma membrane, and then it can have a capsule outside of that. But that inner structure is the plasma membrane. And we know the plasma membrane in eukaryotic cells, like human cells, is a lipid bilayer. It is also a lipid bilayer in bacteria, but in Archaea, it can either be a lipid bilayer or it can be a monolayer where the bilayers have fused. And then the plasma membrane lipids are fatty acids in bacteria, and they're phytonyl groups in Archaea. So they're chemically different lipids in the lipid portion of the bilayer. And we also call the hydrophobic portion, or the portion that's not soluble in water. Okay, so now we're going to talk about the metabolism of prokaryotes, in other words, how they obtain their energy. So we're going to look at two things in metabolism. We're going to look at what is the energy source and what is the carbon source. So for cells to build all of the molecules required to sustain life, they need certain substances called nutrients. Nutrients can be like amino acids, sugars, carbohydrates. Nutrients that are required in large amounts are called macronutrients. Proteins would be macronutrients. Carbohydrates, proteins, these would be macronutrients. And those required in trace amounts or small amounts are called micronutrients. A few elements are considered macronutrients: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Those are 6 elements that are considered macronutrients. And then other elements like zinc or iron, those would be trace nutrients. Now, how do prokaryotes get energy? Remember, I told you to look at the energy source and the carbon source. So the first question is that you ask in determining how to label a prokaryote based on its metabolism is the carbon obtained from elsewhere? Okay, so in other words, does it ingest carbon from somewhere else like we do, like humans do? In other words, it does not make its own food. If the answer is yes, then it's a heterotroph. So if the answer to carbon obtained from elsewhere is yes, then that gives you that it's a heterotroph. Both of these examples on the right are heterotrophs, but if the energy comes from light to convert that carbon into to break down the carbon source into and turn it into energy that can be used in the form of ATP. If the energy came from light, then it's a photoheterotroph. And if the energy came from breaking down chemicals, it's a chemoheterotroph. So humans are chemoheterotrophs. Animals in general are chemoheterotrophs. So are fungi. Plants and algae are I'm sorry. I hope you guys caught that. I didn't make the mistake, but I almost did. Plants and algae are photoautotrophs. So I'm going to put plants over here. And then we have these two strange categories called photoheterotrophs and chemoautotrophs. And those are just unique to bacteria or unique to prokaryotic organisms. So if it makes its own food, in other words, it gets its carbon source from itself, and it gets energy from breaking down chemicals, we call it a chemoautotroph. And then if it still gets its energy from light, but it has to consume the carbon, so the carbon comes from elsewhere, it's called a photoheterotroph. This is just a picture of the carbon cycle, just a review of the carbon cycle.